Hello everyone, this is Group 12. Today, we are going to talk about yeast fermentation process and its application in liquor industry. Yeasts are the most important group of microorganisms contributing to liquor quality in solid-state fermentation process of liquor. This fermentation process involves various stages, including upstream fermentation, downstream separation process, and quality control as well. Throughout these stages, a diverse and complex community of yeast species emerges, each contributing to the quality and characteristic of the liquor. The fermentation of yeast using various plant carbohydrate sources is an ancient human technology that can be traced back to the Neolithic period. The number of strains of yeast are varied in different kinds of liquid production. Although the number of these is not strictly counted, the production of wine and beer is quite different. We then discover some fun facts of production of liquor by employing yeast. The yeast species tended to inhabit different locations of stack and pitch during stacking and liquor fermentation, due to its condition heterogeneity of solid-state fermentation, including the different fermentation temperature and oxygen density in different locations. Moreover, the yeast population was much larger in upper layer than in middle and bottom layer in liquor fermentation, which was in accordance with the profile of reducing sugar consumption and ethanol production. Later on, we will specifically talk about the mechanism of fermentation and production process, includes property, application, and cell physiology and production synthesis, upstream fermentation, downstream fermentation, and quality control sequentially.
Yeast is a collective name for a group of single-cell fungi. The diameter of each cell is about 2 to 6 million, and the length is 5 to 30 million, and some are longer. The individual shapes are spherical, oval, elliptical, columnar, and sausage shape. For the growth conditions of yeast, the optimal pH value is 4.5 to 5, and the optimal growth temperature is generally between 20 and 30. Yeast must have water to survive, but yeast require less water than bacteria. Some yeast can grow in environments with very little moisture, which shows that they have fairly high tolerance to osmotic pressure. Besides, yeast can grow in both aerobic and anaerobic environment. The type of yeast most commonly used in alcohol beverage making is called Saccharomyces cerevisiae. It has a advantage of short growth cycle, strong fermentation ability. It's a large-scale culture and rich nutritional ingredients such as various proteins, amino acids, vitamins, and bioactive substances. It can ferment sugars to produce energy and alcohol. Yeast mostly reproduce through budding in the vegetative state and reproduce sexually under specific conditions. There are three main ways of reproduction: budding, fission, and spawn. This diagram showing a simplified comparison of fermentation and aerobic respiration of a budding yeast cell. This fermentation process mainly consists of two stages. In the first stage, glucose is decomposed into pyruvate. In the second stage, pyruvate is reduced to ethanol. Yeast is mainly used in brewing industry. When brewing, it is necessary to control the conditions to achieve the maximum yield of liquor. First, allow the yeast to perform aerobic respiration, and then budding and reproduction are carried out under aerobic conditions. The yeast can be reproduced in a short time. A large number of individuals can form. Then isolate the air, perform aerobic respiration under anaerobic conditions, decompose glucose and produce alcohol, which can then make liquor.
So I'm with Qi. I will introduce upstream fermentation. In the production of alcohol, fermentation mainly refers to the process of anaerobic respiration by yeast to produce alcohol. In simple terms, a juice consisting of enzymes and a variety of microorganisms is added to pre-treated grain, and the starch in the grain is broken down into glucose by the amylase, and the glucose is broken down into alcohol and carbon dioxide by the alkylase. During the fermentation process, the alcohol content in the mixture increases and then decreases over time. In the early stage, because there is oxygen in the reactor, the yeast can reproduce in large quantities and produce little alcohol. In the middle stage, the oxygen is consumed. The yeast, the yeast anaerobic respiration reaches its maximum, and the alcohol content increases rapidly. In the late stage, the yeast dies and the alcohol is consumed by the variety of microorganisms to produce other substances. Generally, the time to produce alcohol is only half of the fermentation time, and the rest of the time complex reactions occur in the mixed solution to produce acid, ester, aldehydes, alcohols, aromatic compounds, and so on, which are essential for the flavor and taste of baijiu. It's worth noting that during the fermentation process, the temperature of the reactor rises due to the respiration of various microorganisms. So, in order to avoid yeast death, the reactor needs to be fitted with a cooling device. For baijiu, fermentation is a very complex and enigmatic process. First of all, baijiu is a very complex mixture with more than 1,000 components, and there is no precise theory on how to control the proportions of these components. And the production parameters are adjusted mainly through experience. Secondly, the alcohol does not come entirely from the yeast in the jiuqu. For example, the yeast in the high temperature dachu has all been inactivated at high temperatures, and then all the yeast for alcohol production comes from the environment, including the air, the soil, and even the yeast inside the workers, which means that the same fermentation operation in different area will give completely different results.
As can be seen from figure, the next step in fermentation is distillation. Distillation is to put the fermented grains into the traditional still. Using the boiling point of alcohol and water is different. The alcohol in the fermented grains is taken out with steam, purified, and then condensed through the condensing device in the distillation pot, so as to get the wine. After the wine comes out, it is also necessary to pitch in the head and end. The first outflow of wine is called the wine head, and there are some harmful substances in it, which need to be removed. In the middle comes our wine, which are needed. In addition to distillation, we also use membrane separation technology downstream of production. It generally uses ultrafiltration equipment to remove turbidity and improve the clarity of liquor. It also can reduce harmful substances such as fusel oil. Membrane separation can remove the spicy and bitter taste of liquor and increase the flavor. The important function is to improve product quality, prolong shelf life, and reduce production cost. Freshly made wine is not ready to drink. It needs to be aged. That is, stored in a container such as a pottery jar for a period of time. The effect of aging can improve the flavor of the wine body. remove the roughness of the new wine, reduce some low unstable substances and unpleasant odor, make the association degree of alcohol molecules and water molecules better. Through aging, the wine will change greatly. The aroma will be more elegant and delicate, and the taste will be more harmonious and soft. The aging time is different for different wines. And as for lighter aroma generally requires 1 to 3 years. And the requirement of strong aroma is generally 3 years. As for source aroma, its longest is 5 years.
Hello everyone, I'm Lü Hongji. Next, I will introduce our quality control in liquor production. Our quality control system primarily encompasses the following 3 aspects: raw materials, production process, and pollution from packaging materials. First, let's take a look at the raw materials part. The main ingredient used in our liquor production is sorghum, along with a small amount of other grains such as wheat. To produce high-quality liquor, it is crucial to control the starch content and the moisture content of the main ingredients during the procurement. It is also important to check for the presence of impurities and pesticide residues in the raw materials. And in the production process, to produce high-quality base liquor, the environmental conditions of the cellar, such as humidity, temperature, pH, and ventilation conditions, are important. And for the contamination of tools and equipment, we choose stainless steel or regularly inspected and treated ones for rust prevention. And for the liquor that need to be stored in long term, we use uncoated ceramic. And during this period, regular sampling and testing to ensure that the fermentation process proceeds normally is important. The last part is the pollution from the packaging materials, such as we use plastic bottles that may contain adhesives and coatings. This may contain chemicals that can be toxic to humans. These chemicals can gradually permeate into the liquor, especially during long-term storage. Therefore, controlling these sources of external pollution is a critical consideration for maintaining product quality. That's our quality control part of our production system.
It's time for conclusion. To sum up, all the processes are very essential in the yeast fermentation. Alcohol is produced by anaerobic respiration by employing different strains of each cell. In the upstream fermentation process, the concentration of alcohol is determined by the concentration of the oxygen in the container. For the downstream fermentation process, it encompasses the activities of separating, purifying, and processing the desired product or byproduct obtained from the fermentation. There are some mutual technology, such as liquor distillation, membrane separation techniques, and aging. Quality control in liquor production mainly involves 3 aspects: raw material, production process, and packaging material. This is our reference. Thanks for listening and watching.